Okay, we'll get started here in just a second. I thought I was going to be by myself. Yep. All right, good morning. We will get started. Any questions before we get into this last chapter? The end is so near. And from the audience in person, I can tell I got one customer today. That's perfect. I think people are like, geez, why not spring break? What happened to the old times? Okay. Not seeing any questions on Zoom yet. So welcome to you guys as well. I do have a few more customers tuning in live right now, so it's not like it's nobody at all. Okay, so our last chapter, we have made it to the end. We want to talk about nervous system infections. There are numerous things that I think are really kind of terrifying as we move into this last bit. I will say before I start this stuff, yesterday, I feel like I am so close to having the final put together. My plan is to get a study guide posted for you guys. If not by this afternoon, it will happen over the weekend. But I'm pretty confident that you will see something posted, and it will be on the next week's material. I'm trying to keep everything kind of together right where the final exam shows up, so you can see that stuff. Also, with that said, we should have, this chapter isn't tremendous with the amount of images that I'm going to show. We should have time next week to go over that material as well. If you've got any questions or anything like that, be happy to address that. Okay, so moving onward. As I said, definitely some things that I think potentially pretty terrifying. This image maybe is supposed to be of a rabid dog. It looks like a dog that's maybe a little bit upset, potentially. It says it's exhibiting restlessness and aggression associated with rabies. Oftentimes, rabid animals, you might have some signs of like foaming or frothing at the mouth. Hydrophobia, the fear of water, causes excessive salivation, which is kind of odd. But it isn't odd when you think that the virus, the rabies virus, is transmitted through that. So causing excessive salivation enhances the likelihood that that virus would be shed or spread to something else. And we will touch on rabies here in just a second. This image is just to kind of get us started thinking about some of the neurological issues. And rabies is relatively common as far as thinking, you know, not actual infections in people, but common endemic, we might say, in the environment with lots of wild animals potentially harboring this pathogen. And so we think about our own pets, ensuring that they've had rabies shots to protect them and to protect others if they happen to come in contact with a wild animal that might be harboring that. So real quickly, a little anatomy as we start thinking about the nervous system in the first place. So central nervous system, the brain, there's all sorts of possibilities for infection and serious possible fatal consequences. And then we move to the peripheral nervous system, and there are other issues. And we've mentioned a little bit in passing with some of the latency, some of the organisms, virus more so, that may pick up latency or dormancy on some of the nerve ganglion and reemerging later. Shingles, we mentioned, I know, with chickenpox, and it's a potential to think about it here as we're moving through and really talking nervous system. And there are others. Herpes is another possibility with that. So we'll see some of that as we progress along. So this just gives us a cross section, so layers surrounding the brain, and we've got multiple potentials to provide protection, cushioning, and support. We think of this as a privileged site. There are some immune cells present within the central nervous system, but for the most part, with the blood-brain barrier, we try to protect this tissue and not allow, hopefully not allow things in. Obviously, that's not the case, or we wouldn't be talking about some of these potentials here in just a minute. So there are numerous possibilities for organisms to cause serious issues as we will progress through. So here is blow up of some nerve cells and some potentials as we will progress along as far as where we might be seeing some interaction with the specific pathogens. So myelinated neurons, as we're looking at up here, and we also, over here, we can see some of the neurotransmitters. There's possibilities with organisms causing issues at this point at the synapses as well, and that can lend to other problems. Again, we'll hit on this. This is just to give you a quick image of the anatomy. So here we've got in this image two different, hopefully pretty apparent that they are different brains that we're looking at. So the first image right here is normal brain tissue. This was removed at autopsy, and you can compare it to figure B over here, which is another brain, but this individual, not normal brain tissue. They died from bacterial meningitis, and there are a handful of organisms that can cause meningitis, bacterial meningitis. We'll hit some of this here in just 1 second with some of these details. Definitely super serious concern when we think about this, and we'll mention that in more detail as well. So here we could see a lot of pus and then a lot of hemorrhaging that you can see within here as well in the meninges. So all of these little red bits within there, the result of this meningitis. So one of the main ones that we want to think about, and definitely one when meningitis is suspect, this is who we assume we're dealing with until we rule it out, right? Neisseria meningitidis is a very serious pathogen, and it requires immediate attention if we're hoping to save the individual. All right. So these, like I said, cases of meningitis, if bacterial or viral, initially as you present to the clinic, this is what we assume, right? And here you can see these arrows are pointing at Neisseria. Neisseria is gram-negative coccus. Here you can see these reddish or pinkish stained cells, very small. The bigger globs here are white blood cells, neutrophils are what they're saying here. And as I was initially looking at this, I'm like, well, that's really odd. Those don't look like neutrophils to me. But as I look further and see, this is a gram-stained specimen. We're not looking at a blood culture where we are staining that with like Wright or Giemsa stain, where you would see nuclei and all of that. Then it made sense. And it also, I thought, was a little bit interesting as I was looking, scrolling through this morning before I started. These are eukaryotic cells. And so there's no cell walls. It's just plasma membrane, right? And we're doing a gram stain. So it kind of makes sense that we see them staining pink, because when we would decolorize with the alcohol, the membrane should allow the crystal violet to basically just rinse right out of that cell as the alcohol kind of gets in and dehydrates or opens it up a little bit, allowing the crystal violet to basically wash away, like what we would be seeing in a gram-negative cell, like we see up above. So all of this stuff looks gram-negative, right? And ultimately, these eukaryotic cells, gram-stained, appear gram-negative as well. And I think that's kind of interesting. And deep down, as I am thinking about this right now, I know we're not making you answer all of the questions in the lab manual, but there are, I know, with some of the gram staining material, there was a question talking about our own cells and how would they potentially stain if you were to gram stain them. And I think this image pretty much tells you the answer, all right? They would appear negative. So kind of cool. This is a cerebrospinal fluid sample. So a spinal tap collecting the fluid. And when we think about the spinal fluid, this is something that should be a sterile site as well within the body. And so recognition, evidence of some sort of agent within there is indicative that that is the culprit, all right? So it's something that, as we're pointing little arrows at it, sticks out like a red flag or a pink flag, saying we do have a pathogen, right? So, like I said, it is super serious, definitely big issues, and we'll hit some of that with the details here in just a second. But there are potentials for protection. There are vaccine strategies against Neisseria meningitis, the meningococcal meningitis, that provides protection. And some, not all, I feel like it should just be a standard, but many colleges require that students get vaccinated before showing up on campus. And I would think, I mean, even just being on campus sets the stage for issues, but it's more, close quarters. So living in dormitory situations definitely sets the stage to increase the likelihood of some of this. So that kind of might steer you one way or another if you're curious if maybe you should think about being vaccinated. If you're living in a dorm situation, I would recommend it. I'd recommend it in the first place. This next image, so we're still thinking about potentials for meningitis, right? But we're going to shift to a different type of bacterium. One we've already mentioned before, all right? I'm thinking of it more so with pulmonary tract infection. Streptococcus pneumoniae is more common to think of in that circumstance, but it does not have to only plague the lungs. So we've got a couple of images here. Streptococcus, you've probably seen some streptococcus in the lab, I think maybe. Streptococcus is a gram-positive cocci. There are a handful of different types of streptococci. The pyogenes that we've mentioned before that causes strep throat, amongst some other things, is gram-positive cocci that are often big, long chains of these cells. And the Streptococcus pneumoniae is a little bit different. It almost always presents as diplococci, two cells together. This is a fluorescent stain. So they're glowing green, but gram stain wise, it'd be two little purple cocci together, diplococci, right? It's also, looking at them through the microscope, and this isn't the best image because it's glowing bright green, but seeing them gram stain wise under high power magnification, they kind of look lancet shaped, so kind of like a little blade or dagger. This, like I said, not the best because they're glowing and showing the two little cells together. This next image over in frame B is a blood auger plate. And there's some differences between the Streptococcus pyogenes and the Streptococcus pneumoniae as far as their characteristics on blood auger. The pyogenes is beta hemolytic, so it lyses and destroys those red cells, giving you a clearing on the plate. And the Streptococcus pneumoniae is alpha hemolytic, so it only partially degrades those cells, giving you a faint hint of clearing, kind of a brownish, sometimes described almost green-like. This doesn't look green, it's sort of light tan here. The colonies that we can see, the light shining on it nice here, they're nice and shiny, so they produce capsule layer. It's mucoid colonies, you would describe it as. And that also is important with Streptococcus pneumoniae because the capsule layer is a virulence factor. It enhances the capacity for this bacterium to cause disease, to establish by going undetected. And that gives it definitely a leg up to cause infection. So this next one, and I know there are a few as we're moving through here that were figure-wise skipping over. So we will hit, as we move into the table, a couple of other little tidbits. But Clostridium tetani causes tetanus, right? And there are numerous issues with this. In this case, the muscles, we see contraction, we get rigid posture, this episiotomos. As I look at it, it reminds me of like The Exorcist, bending backward, right? You shouldn't be bending that way. There must be demons, right? It makes sense. This is very crazy to see this type of infection. There are other issues. We're not seeing the face. Oftentimes with tetanus, they say lockjaw, and you get kind of this grin. It looks like you're smiling. It looks like you're pretty happy, but you're not, right? There are all sorts of issues, and it's basically the muscles contracting and not able to relax. Its cousin, Clostridium botulinum, does the opposite and causes the muscles to not contract, giving you what they describe as flaccid paralysis. And they use that in Botox injections for facelifts. but there's other potentials where Botox is utilized with certain issues, certain neurological issues where it can cause alleviation. It's also used with excessive sweating, which I think is kind of interesting. A little Botox causes, I'm assuming, the sweat glands to not be overstimulated, maybe. So tetanus, I feel like what this guy needs is a little Botox to alleviate these issues. I don't think that's the case. This next image, Listeria monocytogenes. Here we've got a couple of images, an actual image and then a cycle of it infecting these cells and multiple cells. Really kind of a cool image. This bit here, as we're infecting these cells, I'm looking at this, it looks like a lot of microvilli. I'm assuming this is intestinal epithelial cells, but there are potentials with Listeria to cause meningitis, and that's really what we're focusing on in this chapter. This cycle, like I said, looks more like within the GI tract as you see the microvilli, and it is also an important foodborne illness. So seeing it here, well, it's not surprising to me. So as the cell engulfs it, phagocytizes it, that's basically the process that's happening here as the cell gets into the cell, replicates, and is able to utilize things within the cell. We don't need to go through details with this, but to penetrate into and infect other cells is the idea, and then it basically proliferates through, causing massive cell destruction. So another organism that we want to focus on is Hansen's disease. Hansen's disease, often maybe more easy to understand, is called leprosy. But there's definitely issues when you say leprosy. You potentially have an image painted in your mind. There have been all sorts of accounts of, leprosy and lepers that truly has, painted them in a very poor light in the first place. There's all sorts of stigmas and fears associated with this that are, for the most part, really pretty unfound. So here we could see a nose from an individual with Hansen's disease, and Hansen was the individual that first described leprosy, and it's we're trying to push Hansen instead of leprosy because it's maybe not so much of a stigma associated with it in the first place. So leprosy is Mycobacterium leprae. Mycobacteria have the mycolic acid within their cell walls, making them pretty impervious to gram stain. But the acid-fast staining technique does allow them to show up as kind of these bright pinkish filamentous bacteria. Tuberculosis, a cousin, would look very similar if we were looking at a sputum sample from the lungs of an individual, right? So these, it says here gram-positive bacillus, but I don't know if I truly like that, all right? They shouldn't really stain, all right? With that said, the disclaimer here, if you really had a culture that was kind of getting a little bit old, the cell wall starts to degrade a little bit, and ultimately the crystal violet will get in there and it will be retained. They will look kind of positive, all right? But if we're talking clinical stuff, whether we're talking tuberculosis or leprosy, these specimens should not show up gram stain wise. It should only show up with the acid-fast staining that allows that to visualize them. Last time I put this right in here before the lecture. All right, let me shift over to this text real quick. Um, okay. Okay, hopefully you're seeing my table here. The infections, bacterial. We're still focusing on bacteria, even though we started this chapter with rabies. We're not talking of rabies yet. The bacterial infections of the nervous system. We showed tetanus. We didn't show botulism, but botulism also is an important organism when we think about possible problems. And there are numerous things that we think about, all right? So with infection with Clostridium botulinum, we might think about, some sort of food poisoning situation, or it could be some other issues. So some of the signs and symptoms, blurred vision, drooping eyelids, you have difficulty swallowing and breathing. Again, your muscles aren't contracting like they should. Nausea, vomiting, and often can be a fatal outcome with botulism. So transmission with this is through preformed toxins in the food. Clostridium is an anaerobic bacterium. So these organisms don't like oxygen. We think about improperly canned or preserved food that sets the stage for the bacteria to not be killed as it should. The spores are present. They germinate as that food product gets down to room temperature. And in that semi or relatively anaerobic condition, they're able to grow, proliferate, and release toxins, right? The bacterium potentially will die off, but the toxin is still present, and it's that sets the stage for issues, especially thinking adults. With little kids or with compromised individuals, ingestion of the spores sets the stage for the bacterium to get into the gut, to germinate, and to start growing. And now they become technically a part of the normal flora, especially talking, an infant. And this is not normal flora, but they take over and they proliferate. And then as they are living within the GI tract, they are releasing toxins and can be a serious fatal infection. And this is something like honey as an example. They recommend not giving honey to anybody under the age, I think it's 1, maybe it's 2, I'm pretty sure it's 1. So after that, your immune response is better, but your GI tract is also populated with more organisms to truly set the stage that ingestion of some of these spores aren't able to take hold and become really a dominant player. So use caution with that. And then there's also the potential for it to get in through a wound or injection to get the organism into us as well. There's multiple ways for transmission. Oftentimes, we may think about intoxication, foodborne situations, all right? Hansen's disease or leprosy, we just looked at. Mycobacterium leprae is the organism. It causes a hypopigmented skin. We didn't really see that with the nose on that individual. There's skin lesions and nodules that form. There's issues with this because the nerves are impacted and you lose peripheral function. That causes a lot of these issues. So you see extremities, the sloughing off of things, but it's not, it's the nerve damage that's causing these issues, but it's not as extreme as you may be, the picture you may be painting in your mind, all right? So little different, right? So we think about inhalation of the organism. It's potentially transmissible to us from armadillos, which seems a little crazy, but armadillos are a reservoir host for leprosy, and contact with armadillo does set the stage for you to become infected. So I would use extreme caution. Interestingly, there are ways to try to cultivate this if you suspect it in a patient. A sterile armadillo, one that does not harbor the bacterium, can be inoculated, and you can see if the organism does grow up in the armadillo. I think the kicker with it, if I recall properly, armadillo's body temperature is a little bit lower. Hansen's disease does prefer lower temperature. So seeing areas like the nose, the fingers, or the toes that aren't quite as warm as the core of the body kind of makes sense why you see it in those areas, and maybe the armadillo being a good host. Haemophilus influenzae type B, it can cause meningitis. It's especially important when we think about little kids up to about 5 years old. It can be very serious. It's not something that is huge on the radar really anymore, as long as we're staying current with our vaccine schedules. It is something that we vaccinate against. The Hib vaccine, Haemophilus influenza B vaccine, has been highly effective and made it so that Haemophilus meningitis is very low. Like I said, almost not even on our radar. It's fortunate. And we don't see a whole lot in this category in the first place. So we do see some of the symptoms: nausea and vomiting, photophobia, stiff neck, and confusion. The stiff neck is a common symptom when we start thinking meningitis. It sets the stage for, definitely caution when we're trying to figure out what's going on. And like I said, meningitis by Neisseria is what we oftentimes suspect in the first place. And you can see here the stiff neck right in there. Listeria, listeriosis, initially some flu-like symptoms, sepsis as it gets in and it's proliferating through those cells. It can infect the circulation and transmit it throughout the body. And if it makes its way up into the meninges, we can see a possible fatal occurrence. There's also situations with pregnant women that contract listeria where it may result in miscarriage. So there are definitely some food products that should be off the menu if you are cooking a bun up in the oven. So soft cheeses are definitely things that should be not consumed. Sushi is another thing if it's raw fish. If it's cooked, that's a different story. Listeria, amongst other things, could be possible culprits. Usually it's some other stuff in the sushi, but you still want to use caution. So bacteria ultimately get ingested with contaminated food or water, and there are numerous food products that might set the stage. We have mentioned these guys before with the fact that they really kind of like the cold temperature of a refrigerator and can proliferate and grow under those conditions, releasing toxin that ultimately can set the stage for ill effects. The Neisseria, we started with thinking about these guys and similar symptoms that we had just mentioned with the nausea and vomiting, photophobia, stiff neck, and confusion. The kicker here is this is often fatal. If you don't have very quick treatment with some antimicrobials, prognosis is not good. So direct contact, like I said, close quarters set the stage for kind of ease of transmission with these guys. Next on our list is neonatal meningitis. This is an organism, Streptococcus agalactiae, that can be transmitted to the child in transit through the birth canal. It can be something they contract a little bit later. There are questions, maybe mom doesn't show any signs of this in the first place, but the child gets it. You kind of throw your hands up. We're not 100% with some of the ways that they may be contracting this. There is maybe an enhanced or increased likelihood with premature children to potentially have a greater incidence of infection with this. And that might be, issues where their immune response is not as competent as a child that was full term, making them a little more likely to contract things, maybe, all right? So there's fluctuations, temperature instability, apnea, bradycardia, hypotension, feeding difficulty, irritability, limpness, seizures, bulging fontanelle, stiff neck, the pistachiosis, like back to the picture of the guy with tetanus. And if this is your little fresh hatchling and you're seeing them bent in half, I think you're going to freak out. So it is often a fatal infection. So direct contact in the birth canal, as I said. Mothers get screened for Streptococcus agalactiae. And if they get recognized to be harboring it, because it can be normal flora, it's normal flora in a large percentage of the population. So if they are harboring this organism, we start a little bit of antibiotic treatment to ensure that as they are in the process of delivery, that if contact with the organism occurs, well, basically it doesn't allow contact to occur because it wipes down the population. Streptococcus pneumoniae causes the pneumococcal meningitis. Another same sorts of symptoms. So you can see all of this meningitis symptom-wise, very similar. And back to the point that I was making, you assume it's Neisseria. Tetanus, Clostridium tetani causes, as we showed with the image, the contorted body, spasmodic paralysis, and like I said, lockjaw. It often starts there, and then you see elsewhere. Typically, we think about a puncture wound allowing the organism to get in, and some antimicrobials, antitoxins are also used to deal with this. And we can get this from individuals that donate plasma, and we can extract out some of the antitoxin, some antibody against that. That allows for protection. We also see vaccine schedule, the DTaP, the T in that for tetanus, right? Diphtheria, tetanus, and pertussis are what we think about with that. Okay, I think that's probably sufficient with that. We still have a few minutes left, but that's all of the bacterial stuff right now. And I think that's a good stopping point. So any questions or concerns? Okay, guys, like I said, I will have, when are we taking the final exam? I have the final posted on Moodle. It will open up in 2 weeks. So yes, because today is Friday. So next week lecture, then the following week is closed week, dead week, spring break, whatever you want to call it. You won't be here. The following week is really finals week. So with that, it will open. I think I have it set to open I will check. I think it's Monday of spring break when it starts. Oh man, so many questions. Is it going to get curved at all? I bet you it will. I don't think you should be concerned about a curve before you ever take the test. So other things, I know that I saw more stuff. Where were my chats at? Does it say 3 more? I did see something about Chromebook users. I recommend that you find a different potential for that. You've obviously made it work thus far. So if you have serious issues with that, I guess let me know. But I think there are computer labs on campus that should be open that should allow you to deal with that. How have you made it work thus far? We don't know. All right. There's my chat. Okay, I think I've addressed all of these. Any other questions? Okay, guys. Have a good weekend.